Hi, I'm Associate Professor Lutz Krause, and I'm Chief Scientific Officer at Microba. At Microba, we apply big data methods combined with artificial intelligence to investigate the gut microbiome to improve human health. In the previous presentation, Gene Tyson has shown how we can use these approaches to identify candidate therapeutics and biomarkers from the gut microbiome. In this presentation, I will now show how we can apply the same principles to investigate the gut microbiome for new nutritional interventions, supplements, probiotics, or live biotherapeutics. And I will use anxiety as an example problem, and will show some teaser data. The gut microbiome is still an underexplored organ that presents a wide range of opportunities for the development of novel treatments, such as nutritional products, supplements, probiotics, or live biotherapeutics. At Microba, we apply a human-first, data-driven discovery approach where we mine large gut metagenomic datasets to identify new host microbiome environment interactions and to rationally identify novel interventions and therapeutics. One of our core beliefs is that a data-driven approach can expedite the development of these interventions, and that large datasets combined with high-precision analytical capabilities are needed to identify the most promising signals from the gut microbiome. At Microba, we can discover these new opportunities using our high-resolution taxonomic data, functional data, and nutritional data, and we'll give some ideas on how we use this data today using anxiety as an example. At Microba, we have established a novel gut microbiome discovery platform for the development of new health solutions, which we are now applying together with different consumer health companies. Firstly, we have established one of the world's largest consistently collected gut microbiome datasets, which we call the Microba Discovery Database. We have collected more than 8,000 high-resolution metagenomic samples with very rich associated metadata. This data is consistently collected and analyzed and represents an incredible opportunity for a wide range of discoveries. Secondly, we have developed leading technologies in measuring the microbiome with high accuracy and precision. In the next presentation, my colleague Eleanor will show that our Microba Community Profiler achieves superior accuracy and coverage in analyzing metagenomic data. In the previous presentation, Jean showed that the high resolution of our classifier is critical to identify the most promising biomarkers and therapeutics from the gut microbiome. Finally, studying host microbiome environment interactions from metagenomic data is challenging, given that these associations are complex, sparse, multivariate, and frequently nonlinear. We tackle these challenges using a wide range of analytical techniques, including advanced statistics, cohort matching, and machine learning. As Jean has presented, we have demonstrated that our discovery platform is able to overcome these challenges, and that we were able to rationally select promising novel therapeutic candidates that show strong efficacy in preclinical animal models for UC, so ulcerative colitis. And today I will show how we can use similar technologies, our platform, to identify novel therapeutics or interventions for diseases like anxiety. As mentioned on the previous slide, one of our core resources that we rely on for our discovery is the Microba Discovery Database, a rich microbiome database with extensive metadata. We've collected more than 2,000 metadata points per individual and have metadata on the demographics, medical history, lifestyle, mental health, diet, and physical activity. On the left, we can see some of the more common health or lifestyle conditions that we capture. We have a good sample size for a range of conditions, including gastrointestinal diseases, inflammatory conditions, metabolic diseases, or mental disorders. For example, we have a good sample size for IBS, anxiety, depression, dermatitis, asthma, acne, hypertension, just to name a few. Notably, we have data on a wide range of conditions, many more than the ones shown here. On the right hand is an indication of some of the metadata that is captured for each sample. So for example, we have data on lifestyle factors such as physical activity, age, gender, tobacco use, and others. We have rich metadata on medications like antibiotics usage, metformin, or PPIs. We have extensive data on diet or on other food frequency outputs. As an example application today, I will show how we can mine our extensive gut microbiome data to identify novel interventions for anxiety. Why are we interested in anxiety? 40 million adults in the US are affected by anxiety, and there is clinical need for better management and treatment of patients with this condition. There is mounting evidence that the gut microbiome contributes to anxiety. However, the underlying mechanisms and involved microbial species and functions are poorly understood. Additionally, most previous studies in this area utilized low-resolution 6NeST data. Overall, this lack represents opportunities to comprehensively study the anxiety gut microbiome and to develop new interventions and therapeutics for anxiety. In terms of mechanisms, we don't understand the underlying mechanisms well, but it is well established that there is a bidirectional communication between the gut and the brain, and a number of potential mechanisms have been described. First, microbes can directly interact with immune cells through molecules like flagellin and LPS. The released cytokines can then travel systemically through the body and eventually reach the brain. Second, microbial metabolites such as short-chain fatty acids can also stimulate the production of hormones and neurotransmitters such as serotonin. Thirdly, many microbial metabolites can be absorbed across the intestinal barrier, enter the blood circulation, and then some of these metabolites can then cross the blood-brain barrier. In the Microba Discovery Database, we have a good sample size for anxiety and have more than 300 high-resolution gut metagenomes available for severe to extremely severe anxiety cases and more than 500 healthy controls. In this case, the anxiety categorization was based on the DAS-21 score, a validated survey that we calculated from self-reported information. On this slide, I give an overview on how we analyze the data in the anxiety project. We start with a sample size of more than 600 cases in control. So this was the sample size after applying our exclusion and inclusion criteria. For example, we excluded individuals taking therapeutics like anxiety drugs. We then identified confounders relevant to anxiety, confounders like age, gender, BMI, or stool frequency. We then compared the gut microbiome of cases and controls to identify microbial species, functions, and pathways differentially abundant between anxiety and healthy. And finally, we married this data up with our dietary data to identify associations between food nutritional data and differentially abundant species and functions. This allowed us then to identify potentially nutritional interventions that can shift someone's microbiome to a healthier state. On this slide, we can see that there was an association between anxiety status and microbial diversity, so microbial alpha diversity and beta diversity. So anxiety is generally associated with a reduced alpha diversity. So we see a significantly reduction in richness and Shannon diversity. Now, when you look at the ordination plot here, in this case a UMAP, we can see that there is a trend for clustering of microbial species profiles associated with anxiety. It's not a strong clustering, but there's a trend. And this clustering is indeed significant when testing with undonors. So it has a p-value of 0.003. We found 160 microbial species that were differentially prevalent between healthy and anxiety. To identify microbial species to target for new interventions, we not only considered the p-value of the associations, but also the abundance and prevalence of these species in healthy and anxiety and the gene functions of these species. In the right plot, I showed 10 selected species that are strongly differentially prevalent between healthy and anxiety. 9 of these species are commonly found in the healthy population, but less frequently detected in individuals with anxiety, and 1 species is more frequently detected in anxiety. Interestingly, one of the most significant species that is less prevalent in anxiety is a Faecalibacterium that traditionally is classified as Faecalibacterium prosnitzi in the NCBI taxonomy. At Microba, we use a high-resolution taxonomy based on the GTDB that has divided Faecalibacterium prosnitzi in 13 different species. And in our own research programs at Microba, we have observed that this high resolution is critical to identify the most interesting signals from the gut microbiome. For example, in this case, only 1 of the 13 Faecalibacterium prosnitzi species was associated with anxiety, and this signal would have been lost if we would have used the NCBI taxonomy that groups all of these 13 species into one single species. So the association of Faecalibacterium prosnitzi with anxiety is of interest, as FPRA are the poster boys of health-related species. They are potent short-chain fatty acid producers and have other health-relevant functions. And in relation to anxiety, a rat-based intervention study has shown that Faecalibacterium prosnitzi is a potential therapeutic for anxiety. Notably, this Faecalibacterium was not the most promising species found in our data. For example, species 4 has a much stronger effect size with a high prevalence in healthy individuals. Moreover, species 4 is an uncultivated species, which makes it even more attractive for the development of new therapeutics, such as a live biotherapeutic or probiotics. So this example really demonstrates the promise of a data-driven approach and the need to accurately and comprehensively measure the gut microbiome, including previously uncultivated species. As just described, we're applying a data-driven approach to identify candidate therapeutics from the gut microbiome. And notably, many of the most promising candidates frequently are uncultivated species. At Microba, we have developed several wet lab and in silico methods to expedite the isolation of these species. In particular, we use genome-guided isolation, where we can predict from the genome of the target species the optimal growth conditions. So we can apply this approach to isolate these species, but also to help optimize the growth medium of microbial isolates. In addition to multivariate statistics, we also trained machine learning methods to discriminate between individuals with and without anxiety. And these methods allowed us to identify signatures of microorganisms associated with anxiety status. And furthermore, these methods allowed us to identify higher order nonlinear relationships between the gut microbiome and anxiety status, and also to explore complex multidimensional relationships. So usually when training a machine learning classifiers, what we do is that we split the cohort data in a training set and an independent test set. We then train and optimize a range of models on this training set. We then select the optimal model on the training set and evaluate that one optimal model on the test set. So to train the model, we usually apply a range of data transformations and feature selection methods and dimension reduction like autoencoders, for example. We then train a range of machine learning methods like gradient boosting, deep learning neural networks, support vector machines, random forest-based methods, and others. We then stack the optimal models together using stacking approaches into so-called ensemble models. And finally, we select the one optimal model on the training set. And this one optimal model is then evaluated on the test set. In the case of anxiety, the optimal model was an ensemble model that combined multiple different classifiers, including gradient boosting, distributed random forest, and deep learning neural network. And on the anxiety data, this model was able to predict someone's anxiety status from the gut microbiome with an AUC of 0.88, so highly accurate. We then explored the explainability or feature importance of this model, and the result of that are shown here. So we can see that for classifying samples into anxiety and health status, a range of different species features were included. So the classification was not based on a single feature, but a range of different features. So these models allow us then to make sense out of these black box models or traditional black box machine learning models. They help us to interpret the data, also ensure that the models are not overfitting, and really to understand what the models are learning. The models or this approach then also allows us to identify key combinations of features that are important for disease, or in the case of therapeutics, to support the rational design of microbial cocktails. And finally, these models help us to identify biomarkers for disease. We also identified a range of functions that were differentially abundant between healthy individuals and individuals with anxiety, so in the different function levels, including metapsych, pathways, groups, or RSC numbers, but also on metabolite level. And one of those functions shown here, IPI, that has been previously associated with health, we find a range of interesting metabolites that are associated with anxiety that have previously been associated with health. In the next part of this presentation, I will describe how we investigated food microbiome associations with a particular focus on the anxiety microbiome. The goal of this analysis was to identify food items that can potentially shift anxiety-relevant microbial species towards a healthier state. This is ongoing work, but I can already show some interesting results today. We first identified associations between food intake and the abundance of species differentially abundant in anxiety using univariate statistics. This did identify significant associations. However, the associations identified by univariate statistics were low, with only a low amount of variance explained and a low R-square value. We next modeled the abundance of the top associated species with multivariable methods, in particular lasso regression. With lasso regression, food intake could explain much more of the variance in relative abundance of these species, with R-square values of up to 0.68. So by modeling the abundance of these species by the entire food intake data, we could explain more than 50% of the variance in the abundance of these species, which is actually quite remarkable. So in essence, these results show us that food microbiome associations are complex, as expected, and that we need to analyze the data with methods that can handle this complexity, including methods like lasso regression, partial least squares, or machine learning methods. The heat map on this slide shows the coefficients learned by the lasso model, and as we can see, as expected, the associations between food and microbial abundance are complex and multivariate, and multiple food items contributed to the model for each of the 10 species, food like grapes, yogurt, beef, and other items. We can also see that, as expected, different food items were associated with different species. In addition to lasso regression, we also trained a machine learning method to predict the abundance of our top associated species. And these models were deep learning neural networks. Also, these models were able to explain a large proportion of the variance in the abundance of these top 10 species, with an R-square value of up to 0.65. In the middle plot, we can see food items that were learned by the model to predict the abundance of species 4, and as we can see, also in this case, the relationships between food and microbial abundance are complex, with multiple food items contributing to the model. One advantage of machine learning methods is that they can learn higher order nonlinear relationships. So for example, in this case, the top associated food item had a nonlinear, almost cubic relationship with the relative abundance of species 4. This is just the start of the discovery process for developing interventions. We have run a proof of concept study identifying key differences between the healthy and the anxiety gut microbiome, and have identified food microbiome associations. The generated data can now inform the development of gut microbiome targeting interventions to treat anxiety. So far, this is just the beginning. We have generated some proof of concept data, but there is still much more left to explore to ensure the most promising signals are targeted. And this program is now available for partnership and co-development through its next stages. As an outlook of the next steps, we would like to further study the anxiety microbiome to identify the key microbial factors that likely contribute to anxiety, to select the microbial species and functions to target with new interventions. We can then identify food items or nutritional interventions that modulate key microbial factors associated with anxiety and shift the anxiety microbiome towards a healthier state. Using our comprehensive dietary data, we can also look beyond whole foods and can investigate micronutrients, macronutrients, fiber types, or polyphenols to identify different types of interventions to modulate the microbiome. So together with partners, we can develop new food or nutritional products to target the microbiome, new supplements or next-generation probiotics or live biotherapeutics. And as Jean has presented, we have a microbial isolation characterization platform which achieves great success in isolating even previously uncultivated target species. As a summary, in this presentation, but also in the last presentation of Jean Tyson, we have seen that there is a huge untapped potential for developing new interventional products targeting the human gut microbiome. In our proof of concept study on anxiety, we have identified multiple microbial species and functions that are differentially abundant in individuals with anxiety. We have also found that these species and functions are associated with the intake of different food items. This information can now help us to develop new food or nutritional products to improve the gut microbiome of individuals with anxiety, or to rationally select candidate microbial species for the development of probiotics or live biotherapeutics. So in our proof of concept study on anxiety, we have identified a number of microbial species and functions that are different in individuals with anxiety. We've also identified nutritional interventions that potentially can modulate these species and functions. This is ongoing, and we are now looking for a partner to progress this work and develop them into new products. As mentioned earlier, Microba's discovery cohort covers a broad range of health conditions, and while in this presentation I focus on anxiety as an example, we also observe strong microbial signals for a range of other health conditions. For example, we observe strong signals for a range of gastrointestinal diseases, metabolic conditions, cardiovascular diseases, atopic disease, and mental disorders. Also, these conditions can be further explored using our technology to identify new gut microbiome targeting interventions that improve health. Thank you for your attention.